on track.
Sure.
I may not be able to ask that because if we say yes, then have to All right, we'll go ahead and get started. Welcome back. I'm James Peterson. I am the administrator for Northwestern's Center for Chromatin Nanoimaging in Cancer. We got our technology development talks today, starting with Hao Zhang from Northwestern, open source optical detection and data processing modules for spectroscopic single molecule localization microscopy.
Thank you. So this is a first of many in the future, talking about how we develop technology and how we are trying our best to share the technology with researchers and inside and outside the campus. This is a real collaborative effort. We work with Vadim's team, Daniela's team, chemistry, biology, across all over the places. So I only show the optical design today, but the project is much bigger than that. So here's the outline. I will talk about the fundamental of super resolution microscopy. To be more specific, is a single molecule localization microscopy. And I'll show very briefly a few examples of why we think it's powerful and can contribute to the whole field. I'll explain the optical design aspect, and I'll show our effort to make it more accessible in terms of data processing. A very brief history of the super resolution aspect is a 2014 Nobel Prize. Multiple researchers share the actual award, but there are more actually contribute to the field. Among the more that I actually contribute, did not being recognized by Nobel Prize, it's Howard Hess and then Hao Zhang. Our work follows the localization microscopy. People use PALM or people use STORM. So here's the fundamental principle of localization microscopy. Assuming there are 5 molecules, they're so close to each other because of the diffraction limit, which is the inverse proportional to the numerical aperture, proportional to the wavelengths, and there is a physical barrier that you can't overcome. So these 5 are so close to each other, we know they are a few molecules, but we don't know where they are. Now, if there's a magical way, it's actually the photodynamic processes that statistically, if we only let one generate fluorescence photon, all the other 4 remain dark, quote unquote dark. If we're confident that there is only one molecule light up, we can ignore the actual size that dictated by the numerical aperture. We forgot about the size, and then just take the center using a Gaussian fitting and a multiple type of fittings. So keep the center. And then in the second image, a different one light up. In the third image, yet another one light up. And then we just repeat the process until we are pretty much confident saying that the majority of the molecules light up and we form an image. This is not an intensity image, so it's very different from confocal or epifluorescence image that reflects the local concentration of molecules. This one is a point cloud. It's a single localization after the calculation. So these are all great, Nobel Prize, but what if they're different? And we want to know not only where they are, but what they are, right? A straightforward way is we'll add dichroic mirrors. And the dichroic mirrors separate the fluorescence spectrums according to their band. And some are passed, some are reflected. Fantastic idea, but what if the fluorescence molecule are actually highly overlapping? Is there a way to overcome that? And for lots of applications, we actually want the fluorescence molecules to be similar to each other because of multi-molecular labeling. If they're too different, it's really challenging to label them in the same cell with multiple contrast. So there are some benefit of actually doing overlapping spectrums. So here's the idea that we contribute to the field. Here's the fluorescence molecule through the imaging process. If we add a component, the component is a dispersion component. It can be a grating, can be a prism, or it can be a combination of grating and prism or prism assembly. We'll separate the photons into two beams. The first beam remain the spatial location. The second beam will disperse the fluorescence photon to have a spectrum, and then add an imaging setup, so an imaging lens, simplify that with a camera. And these two we capture by the same camera at the same time. Because this is a stochastic process, we have to capture that at the same time. Otherwise, there's no way back. It's a random process. So what we have a zero-order image reflects the typical location of the molecules. And we have the simultaneously acquired first-order image reflects the corresponding emission spectrums of every single molecule. Numerically, we apply different algorithms to combine them. Now we know where they are and what they are. So that's the general concept. There are, of course, pros and cons. What you're looking at is the first implementation we did is a reflecting-based grating. So this is a turf microscope. We routed the detection beam through the reflection beam, through the reflection grating, and then using a focusing. Now we were using a curvature mirror to reflect it to an EMCCD. So on the left side, this is a stochastic emission of the localization. On the right side, these are the associated fluorescence emission spectrums. And we take frame by frame after the calibration. We'll try to publish the calibration process in the very near future. After the calibration, run through the model. So this is not just with divided photons. We introduce a new term, spectral precision. All we have learned so far are spatial localization precision. Now we have to introduce a spectral precision. And these two fight against each other because they share the same photon budget. So assuming we figure out all this, and here's one demonstration. These are the three-color images. That's the point I want to show. They're essentially the same if you apply a dichroic mirror. There's no way to separate them. But because we know the full spectrum, digitally we can reconstruct that. And that's the final image, three-color staining. And the take-home message was the number of, as long as it's chemically feasible, biologically feasible, the number of fluorescence molecules are actually unlimited. And there's a story behind that. We started the project actually in the same year as the first paper published. We published in 2016, first paper published by a core group from Berkeley. They published 1 year ahead of us. It's not because of optics. We don't know how to do the staining. We actually had to send the sample to Genelia Farm, and Leon's team helped us to stain. Otherwise, we won't even to publish any of that. One application I want to talk is we want to investigate the co-localization of microtubule and mitochondrial. Specifically for mitochondria, we're trying to quantify the individual mitochondrial volume, aspect ratio, and protein density at the outer membrane to evaluate the healthy level of mitochondria. So these are the healthy mitochondria. These are the mitochondria the cell going through apoptosis. So mitochondria going through the fragmentation process. We quantify the individual mitochondrial, quantify the aspect ratio, the total volume, and individual volume, do a statistics, and hopefully the statistic will show up differently when the cell is going through pre- and apoptotic process. This is the overview. Multiple projects are going on. The center part is a primary culture retinal neurons, and we're trying to evaluate the likelihood of the neuron to successfully integrate into the photoreceptor when the reprogrammed retinal neuron is being implemented to the human eye. So this is the cell we received from Jeff Goldberg from Stanford. On the left side is a paper on the revision. We trying to investigate the functionality of nuclear pore of the stem cell and the cancer treatment during the treating process. What's the dynamic of the transport across the nuclear membrane? And of course, working with Vadim and Daniela's team, we do quite a few chromatin imaging and trying to correlate that with a spatially resolved genome sequencing. So back to the optics part. We're not the only one working on this family of technology. There are multiple papers published by multiple groups around the world. So I'm trying to walk you through the processes. this is our initial design. And you see here, this is a grating. And grating separate, now it's transmission grating. The grating separate the photon into two beams, the spatial beam and the spectral beam. And we integrate that. 1 year before us, the team from Berkeley, they use a beam splitter, separate the photon into two beams. And for one of the beam, they actually use a well-controlled prism. And it's actually achieved the same goal, but just use more component. Later on, there are groups reporting the 4-pi detection. 4-pi detection actually had benefit. Why? One detection going to the spatial, one detection going to the spectral. Maximize the photon detection. So it's basically double the photon budget. The downside is 4-pi is by definition, you have to use single object. You cannot use anything thicker than the working distance of the high magnification of objective lens. We're trying to maximize the photon utilization. So we did a try of the so-called symmetric dispersion. So in other words, we don't have a spatial image anymore at all. Both images were spectral image, but they are symmetrically dispersed according to the imaging or the incoming beam direction. We can virtually reconstruct the spatial image. I'll show one example. And this one, Wei Hong from my group from Northwestern Center will show our history of iterating the dual beam, the dual wedge prism assembly. Everything is trying to boost the photon utilization. I'll just give a hint. So for this one, how many components we're going through? 1, 2, 3, 4. 4 components at least. And if you count the refractive index of air and glass, 1 to 1.5, that's 4% loss without coating. And the transmission grating has 25% loss, regardless how hard you work on that. So with all these components add up to each other, this is a significant photon loss. Doesn't do anything. But if we integrate everything into a piece of glass, there's one interface, there is a second interface, that's it. So we're trying to minimize the number of interfaces, and also there's a benefit of how to integrate that. So we talk about the optical design and how to do 3D imaging. There are two mainstream 3D imaging implementations. On the left, in the middle here, was first published by the Xiaowei's group from Harvard. They introduced the astigmatism by adding a cylindrical lens so that the distortion of the points by function are non-symmetric above or below the focal plane. By analyzing the non-symmetric distortion of the points of function, we can back calculate the depths. Another very popular design is the bipolar design. Bipolar design is there are two imaging planes by two different optical imaging passes, and the camera is actually in the middle of that. By distorting the imaging plane, we can back calculate by comparing the relative size of the same points by function. But there is a calibration process associated with that. So King He, he contributed significantly to the initial optical design. He's now a faculty back to Korea. What he did is we had the optical, the zeroth order for spatial beam. We have the first order for spectral beam. In order to achieve a bipolar design, he actually added a pair of mirror. Of course, it's a highly coated, high reflectivity mirror, so that we have the bipolar, similarly to biplane distortion of the points by function, but we can only do along one direction because there's a dispersion axis corresponding to x, but it's actually used for lambda. Using the same calibration, so you see this two curve calibration, we can calculate the z location, so that's the 3D. And this is what I'm trying to explain, the so-called virtual spatial image. On the left is a spectral image, on the right is a spectral image, but they're symmetric to each other. So we can calculate the center point of these two spectral images, reconstruct the spatial image. What's the benefit of that? All photons are used for both spectral and spatial. So there will be a quantitative measure to see how much improvement we can achieve by using the symmetric as compared to the non-symmetric. Now, because we have two beams to begin with, introduce another pair of mirrors, we can achieve biplane. So I want to get it started why we're doing the DWP design, and Wei Hong will show a more detailed explanation of that. As I mentioned, in order to make it a lot easier to be installed, and a lot less interface to cause the photon reduction. We design a assembly. The first beam splitter is by choice. We separate that by 3 to 7. And then going through a 2-prism assembly, the 2-prism we actually design, we control the refracting index, we control the angle, so that there's an on-axis dispersion. And then because of the optical path lengths are different, the 2 focal plans are offset by a controllable path length. If we put a camera in the middle, we have a 2 biplane. So at the end, what we have is, forget about all these complicated assemblies of mirrors, of gratings, of focusing mirrors. It's basically a small component, as small as a quarter, and insert into assembly. So this is a commonly used inverted microscope. And that's actually what the magic, the DWP is going to do. If we zoom into this area, the DWP is insert to the side port. There's no alignment. There's no calibration. Sorry, there's no maintenance. Insert there, screw it on, lock it, add a EMCCD. Every microscope can be achieved as a spectroscopy, single molecule localization microscopy. Now, we finished the optical design, and we continue to improve that. Janelle, she graduated 2 years ago. She championed the project that we're doing ImageJ, because we know how to do it, but not everybody is actually are capable of writing his or her own project. And there are more and more groups actually either developing or adopting the spectroscopy single molecule. So she created a project on GitHub, and these are the functionalities. And I'm sorry, it's a little time. I'll fly. So calibration, visualization, and processing. These are the key processes. So she showed that the program is adapted to both grating and prism based. The spectral calibration doesn't have to be linear. Grating is linear. Prisms are nonlinear. So we're using third order of polynomial fitting to calibrate the spectral. And then trying to analyze different spectrums based on the 2D imaging as an art is also a science. It's all signal-to-noise ratio limited. So we're trying to develop multiple ways to do it. One of the ongoing effort, and will be integrated into the program, is the machine learning based. So in other words, I'll bypass. We're trying to do the spectroscopic classification based on machine learning. One of the clear benefit is in quite a few biological processes, one molecular concentration can be 1,000-fold higher than the other molecules. If we don't do a careful, precise calibration of the spectral classification, the low abundant molecule will totally disappear, may not 100% reflects the actual biological processes. So I'll stop here. We developed the spectroscopy single molecule localization microscope. We're trying to push the hardware and software. We'll continue to improve that, and you're welcome to try. And here, it's for free for now. we have 10 units arrive a month ago. 6 are claimed. There are 4 still. All right. Okay. Thank you.
Nice work, Hao. Can you hear me? What is the spectral resolution, the smallest nanometer difference that you can see in terms of?
All right, the question is, what's the spectral resolution? So the spectral resolution is a trade-off between the spectral precision and spectral resolution. The high spectral resolution means we have to spread the photon into more pixels, where each camera CCD element will see less photons, and that's the noise limited process. So it depends on the actual need. If you are talking about quantum dot, the photon budget is unlimited. Go as high as you want. But if it's Alexa floor, so the rule of thumb is 5-ish nanometer. So
So does that mean that also your spectral resolution will also change from region and region of the image?
It is. It's a photon-limited processes. The beautiful aspect for spectroscopy-based imaging is, I really want to champion this. Without spectral information, there are multiple work published in high-profile papers trying to cascade number of frames. So typical frames are 50,000. The camera keep exposure for 50,000 times, and then all 50,000 images are independent from each other. What a waste, because one molecule will keep blinking at frame number 30 to number 45. Now here's the spectrum. We can combine different frames, and it's actually numerically boosts up the spectral precision.
Thank you.